in these differences, you can understand that what can be the structure of dicot plant and what can be the structure of monocot plant, because externally when we observe then we can easily identify them with the help of these structures. In the dicot, the root is thick and it has a proper system of the taproot. It arises from the radicle and it is called the taproot. But in the monocot, root is fibrous and it is having very thin structure and they are not very deep inside the soil, which is called adventitious root. So here we can see, I'm sharing another page with you. Here we can see the leaf of dicot. This is dicot, this is monocot. Leaf of monocot with parallel venation, pointed leaves, right? Here we can see the midrib, and from the midrib the veins are arising and they make a network which is called a reticulate venation or network-like venation. Leaf stalk is also present. The leaf in the monocot stem is erect and node and internode are prominent and leaves are directly arising from their stem. They are directly attached. They don't have any leaf stalk. In the dicot, stem is branched and it has furthermore branches where leaves are arranged like this. So it's a branch structure. The flower of the dicot may have 4 or 5 or the multiple of 4 or 5. Here the 3 or multiple of 3, like 6, like others. They have like this. Three or multiple of 3. The roots in the dicot are thick root. There is a radicle of the seed. From radicle the root arises. And the primary root, the first root which arises is known as primary root. From the primary root, the branches which arise are known as secondary roots. From secondary roots, the roots which arise are known as tertiary roots. From tertiary roots, root hair arise. At the end of root hair, root cap is present. Root hair or root cap. And after root hair, root cap is present. Root cap is present. In these plants, roots are fibrous and adventitious roots, and they may also arise from any part of any other part. Like they may also arise from here. They may also arise from any other parts. They are known as adventitious root. They will not arise from the radicle of seed. When we talk about the root, stem, leaf, flower, and the seed in these plants they have only one cotyledon. In these plants, leaves have two cotyledon, or the you can say here the embryonic leaf. Okay. Here I'm going to share with you the other thing. From your book, you can see parenchyma. Here they are showing you, I have explained this. What is the difference in the dicot and monocot? Dicot and monocot, they have told you here. Then we talk about the Venn diagram first. What is Venn diagram? Venn diagram means the diagram which is mentioned here like this, the TS. They are going to tell us about the fact that in the plant diagram where the tissues or cells are mentioned, we are not going to make individual cells. We just make the lines through the lines we are going to mention the different type of tissue through the lines we mention the tissues. So outer layer is the epidermal tissue, we make 2 lines. After that we can say that the vascular bundles are present like this. Vascular bundle, in the vascular bundle we can make line to separate the xylem and phloem. So we just make line to mention the arrangement of tissues. We don't make individual cells in the diagram of a tissue. Like if they have made the leaves, let me further change the page. See, this is the structure of the TS of the stem, right? In TS of stem, you can see this is the stem diagram of the cell which are visible and the tissues which are visible inside the stem. But if they ask you to draw the diagram of this, you can see this is the area where a different tissue is present. This is the area where you can find a different tissue. In this tissue, you can see the cells are different from here to here. You can also see one of the different layer of another cell. So in this way, you have to mention these cells because in this you can see internally they are different light blue colored cells. Externally they have purple shade cells. So wherever the cell will be changing, the formation of cell will be changing. It shows you a different type of tissue and you have to mention this tissue with the help of line. So each line for one tissue. You are not going to make individual cells. If you make a cell, you are not going to get marks on this. Then we come towards the next one. In the next diagram, they are showing you this example here. You can see the different tissues are present, but they are just showing you the lines where the change in the diagram is visible. Here you can see the leaf, TS of leaf, and in this TS of leaf, we can see the outer layers. You can see here. Or if I zoom it out like this, you can compare the diagram of these two leaves, right? So this is the way you have to draw the